As we have seen before, organismal aging is highly complex and varies from organ system to organ system. One problem with the brain as an aging organ is that it itself is highly varied, with many extremely specialized areas. For example, there's the hippocampus. As we just talked about, it is the region where adult neuronal stem cells were discovered. There is the striatum, the region of motor control of the body, and so on. So many different tissues exist with different functions that are affected by aging in different ways. Other than being made from similar cells, such as neurons, astrocytes, or oligodendrocytes, these regions have very little in common and, in fact, respond to aging very differently. The investigative approaches to brain aging therefore need to be similarly diverse. The National Institute on Aging, or NIA, operates under the umbrella of the National Institutes of Health, which is the nation's primary funding agency for biomedical research. Roughly 60% of the NIA's budget for research is earmarked towards a single disease, Alzheimer's disease. This is more than all other NIA programs combined. In contrast, only about 13% of the NIA budget is dedicated for funding of basic research into the biology of aging. Currently, approximately 5 million Americans suffer from Alzheimer's disease, a number that is expected to rise to around 13 to 14 million by 2050. Along with the rising incidence numbers, treatment costs are similarly expected to skyrocket and actually dwarf research expenditures. And that despite Alzheimer's disease being the number one priority at the NIA. AD is truly a disease of aging, as it is the most common cause of dementias in people 65 years or older, and it is the sixth leading cause of death in the elderly. AD is characterized by progressive memory loss, loss of impulse control, and personality changes. Although disease etiology may be variable, it also is a very perfidious disease. Long before even mild symptoms of mild cognitive impairment can be discerned, the disease may already be at work. By the time full Alzheimer's disease has been diagnosed, the disease is incurable, it is progressive, and nothing can be done anymore. Current treatment largely is aimed at alleviating symptoms. During the course of the disease, massive cell loss occurs in the brain, especially within the hippocampus, leading to large lesions and overall brain shrinkage. At the cellular level, amyloid plaques and neurofibrillary tangles become prevalent. These neurofibrillary tangles are associated with a protein called tau. Tau is a small protein that binds to microtubules. Microtubules are proteins that are part of the cellular cytoskeleton and help maintain cellular shape. Especially in neurons, microtubules also have a function as a sort of a cellular transportation superhighway. Along this highway, important cargo gets distributed throughout the cell. In a normal, healthy cell, tau binds to microtubules and helps stabilizing them. Certain stimuli, however, pictured here as inflammation, often thought as a major contributor to the aging phenotypes, lead to the hyperphosphorylation of tau, which changes how it is associated with microtubules. This destabilizes the microtubules and eventually leads to their complete disintegration and destruction, only to be replaced by clumped together tau protein. These tau tangles kill neurons, but unfortunately they may themselves survive this process. Phagocytic cells then attempt to remove those leftover tangles, and that, of course, in turn leads to an inflammatory response as part of the phagocytic activity of these cells, and the vicious cycle continues. More inflammation leads to more tau hyperphosphorylation, and so on and so on. Many researchers believe that inflammation is in fact a key driver in AD pathology, especially when it comes to tau tangles. Unfortunately, anti-inflammatory drugs thus far have failed to perform well in inhibiting AD disease pathology and progression. The other common phenotype of AD, next to fibrillary tangles, are senile plaques, or also called amyloid plaques. These plaques are composed of a small peptide called amyloid beta, or A-beta. A-beta is generated from APP, the so-called amyloid precursor protein through the activity of enzymes called secretases. APP is a transmembrane protein with possible roles in synaptogenesis or even the regulation of dendrite outgrowth from the main cell body. It is hypothesized that APP and its regular cleavage products play crucial roles in the regulation of brain plasticity by controlling the number of connections that neurons can make between themselves. Differential cleavage by these various secretase enzymes may produce a variety of fragments, some of which may even oppose normal APP function. When APP is acted upon by beta and gamma secretases, the small A-beta protein is produced, which then can aggregate into highly insoluble polymers. The exact mechanisms how A-beta plaques form are still unclear, but it has been suggested that A-beta functions similar to a prion. A prion is an infectious protein, a small, incorrectly folded protein that can impart its own improper structure onto other proteins, therefore infecting them, if you will. These infected particles can then go on to form polymeric fibers and insoluble plaques. How A-beta plaques lead to neuronal cell death is currently a very active area of research. One possibility is that plaques may activate cell receptors called death receptors, or short DR, which in turn activate cell suicide pathways. That kills the cell. A-beta plaques may also induce inflammatory responses, which of course we have seen before, leads to tau hyperphosphorylation, tau tangles, and so on, really furthering disease pathology here. A-beta plaques may also induce inflammatory responses, which, as we've seen before, in turn may lead to tau hyperphosphorylation, neurofibrillary tangles, and so on. The vicious cycle simply continues. Another important disease of the aging brain is Parkinson's disease, or PD, a disease characterized by involuntary movements and tremors, but also memory and personality changes. PD sufferers show progressive cell loss within the basal ganglia. It is thought that PD originates from mitochondrial dysfunction, which we will discuss further in a later chapter. Similarly, ALS, or Lou Gehrig's disease, is caused by cell loss that affects mostly motor neurons, resulting in difficulties with motor control, especially a stiffening of muscles, making movement very, very difficult. Finally, prion diseases are another example of neurodegenerative diseases that lead to cell loss. As discussed earlier, prions are small misfolded, infectious proteins that force their own misfolded state onto other proteins, which usually leads to insoluble plaques. So these are just a few examples of neurodegenerative diseases, each affecting a particular brain area or function. What many, if not all of these diseases have in common is the accumulation of insoluble aggregates or plaques that are highly associated with neuronal cell loss. Which specific neurons are lost largely determines the phenotype of the disease. Another disease that affects the aging brain is cerebrovascular disease. It is the third most leading cause of death in the US after cardiovascular disease and cancer. Essentially, it is the hardening of blood vessels in the brain and deposition of insoluble plaques within these vessels, often high in cholesterol and saturated fatty acid. This reduces vessel diameter and blocks blood flow. Due to lack of elasticity, vessel diameter can no longer be adjusted, thus the effective brain area is deprived of blood and therefore oxygen. As a consequence, tissue starts dying. An infarction or stroke is the consequence. During aging, elastic proteins that line the blood vessels become cross-linked and thus less elastic. Due to this lack of elasticity, vessel diameter can no longer be properly adjusted. Thus, the affected brain area becomes deprived of blood and therefore of oxygen. As a consequence, those tissues start dying. An infarction or stroke has happened. In addition, often heart function declines with age as well, which further decreases blood delivery to the brain and further increases the risk of strokes. Depending on the size and location of the infarction, various behavioral, motoral, memory, and other phenotypes may manifest. The incidence of these diseases is higher in the elderly. The incidence of all the diseases discussed in this section is higher in the elderly, in part due to their rather long incubation time. But just like with many diseases of aging, they're probably more symptom rather than cause of aging. Check your understanding. Describe the disease pathology of Alzheimer's and Parkinson's disease. Explain the current molecular understanding of AD and PD. Explain mechanisms of cerebrovascular disease. Describe interventions that have been successful in improving normal and pathologic brain aging.